Assalamualaikum warahmatullahi wabarakatuh and good morning everyone. Welcome to the Biology Professor Lecture Pusat Asasi UiTM. Thank you for this semester. My name is Masita Abu Kasim and I am your moderator for today. First and foremost, I would like to welcome our guests yang berusaha, Profesor Madya Dr. Norrizah Jaafar Sidik from the Faculty of Applied Science UiTM Shah Alam. And warm welcome to our dearest biology lecturers and beloved students. Praise to Allah Subhanahu Wa Taala, we have been graced by the chance to gather in these momentous occasions. Before we move on, let us begin with the recitations of dua that will be led by Muhammad Faris Wajdi bin Yusri. Silakan. Muhammad Faris. Mana Muhammad Faris? Please. Okay. I think I continue. Okay. Thank you. Okay. With us today is Professor Madya Dr. Norrizah Jaafar Sidik. She will give a lecture on plasma membrane transport. We are privileged to have Dr. with us today. Before we start, let me introduce a little bit of her education background. So, Dr. Norrizah currently works at the School of Biology, Faculty of Applied Science, University Technology Mara Shah Alam. And she holds an undergraduate and master degree from University Malaya and completed her PhD degree in plant biotechnology from University Putra Malaysia. She had an experience teaching a research methodology, plant physiology, biosafety and biosecurity, contemporary biology, biodiversity, botany, animal physiology courses for undergraduate and also postgraduate. She also supervising local and international postgraduate students from various countries. She is a member of Excellent Entity Agriculture Biotechnology Research Group UiTM, International Society for Horticulture Science, Natural Product Society, and Malaysia Society Plant Physiology. I am sure Dr. Norrizah can share many of her experience with us today. Okay. To all students, if you have any inquiry, please do ask questions during Q&A sessions later, or you can write your question in the chat box. Okay, without further ado, now let's get started with our professor lecture for this semester. And with that, I would like to invite Dr. Norrizah to give her lecture. The floor is yours, Dr. Norrizah.
All right, okay. Thank you very much, Dr. Masita Abu Kasim as the moderator. Right, so I will start the lecture now. Okay, so I will share the slides. Okay. Okay, hopefully all of you can see the slides, right? On the plasma membrane transport. Yeah. So if you look at the pictures here, okay, generally, okay, these are the plasma membrane. Okay, so I show with the cursor here, right, the plasma membrane, and in the cellular biology, the plasma membrane transport actually are the collection of mechanism that regulates the passage of solutes. So the solutes, it can be ions, small molecules, or the small molecules, right? And it will move across or through the plasma membrane here, which are the lipid bilayers, lipid bilayers, and it has the protein embedded in them. So this is only a general view, right? So I will go to the next slide on the learning outcomes. So if you look at these slides, it is listed 5 learning outcomes. Okay, so at the end of the course, students should be able to list the components of plasma membrane and their functions, describe the structure of the plasma membrane and the functions of each of its components, and explain the role of membrane protein, explain how membrane structure results in selective permeability, explain the various transport mechanism across the membrane. Okay, so these are the 5 learning outcomes. Okay, now we look at the membrane components and its organizations. Okay, so if you look at the figure on the right side here, right? So it shows the plasma membrane, okay? The components of the plasma membrane, where you can see the protein channels, the lipid bilayers, the carbohydrate chains, okay? And you can see here, this is the outside of cell and inside of the cell, the cytoplasm. And where can you find this plasma membrane? Yeah? This plasma membrane can be found in the prokaryotic cells, such as the bacteria. Yeah, so you should know what are the definition of prokaryotic cells. Yeah? And also the plasma membrane also can be found in the eukaryotic cells. Okay, by now students should know the difference between the prokaryotic cells and the eukaryotic cells. Okay, now we look at look back at these slides, right? The plasma membranes. Yeah. So if you look at the plasma membrane, it looks like a sea lipid, yeah, with an iceberg floating on it. Yeah. So the iceberg is like, these are the proteins, right? Okay, so the plasma membrane, it provides protection for a cell, for a cell, and it also acts as a barrier. Okay, so it provides a fixed environment inside the cell. Okay, so plasma membrane, it can transport ions, water, nutrients in and out. Yeah, and the plasma membrane regulates not only what enters the cell, but how much of the ions, the water or nutrients comes in. Okay, so and it also exhibits selective permeability. So what does it mean here? Yeah, selective permeability, it refers to its ability to differentiate between different types of molecules. Okay, so it only allowing some molecules through while blocking other molecules. Okay, so it only allows certain substances across. Okay, and if you look at the components here, the most abundant lipid in the plasma membrane is the phospholipids. Yeah, and the phospholipid are amphiphilic molecules. Okay, so the word amphiphilic molecule means it containing the hydrophilic regions and the hydrophobic regions, right? So if you look here, these are the two phospholipid, one layer and the other layer, right? So it has the hydrophilic head here, yeah. And the hydrophilic heads of phospholipid is a membrane bilayer where it facing outward, yeah. So it facing outward, it contact with the aqueous or the water fluid both inside and outside the cell, right? And water is a polar molecule and it is readily form the electrostatic charge interactions with the hydrophilic heads. Whereas if you look here, these are the tail of the phospholipid, which are the hydrophobic. Yeah, or what we hydrophobic, it refers to water fearing. Yeah, so the part of phospholipid consists of its long non-polar fatty acid tails. So the fatty acid tail can easily interact with other non-polar molecules. Okay, and but they interact poorly with water. All right. So this is the components and the organizations of the plasma membrane. So now we look at the structure of plasma membrane. Okay. So before the invention of the electron microscope, okay, the researchers, the biologists, yeah, scientists, yeah, they have identified the plasma membrane, but there is no direct evidence, yeah, and they have proposed, yeah, or they have hypothesized different models, right? And after the invention of the electron microscope, okay, many scientists contributed to the development of modern membrane model. Okay, so the fluid mosaic model, okay, as you can see here, right, is widely accepted model, right? So these are the currently accepted model for the structure of plasma membrane. It was first proposed by S. Jonathan Singer and Garth Nicholson, yeah, in 1972. Okay, so if you look here, the fluid mosaic model, the plasma membrane is a mosaic of components, yeah, where it composed the phospholipid, cholesterol, and proteins, and they move freely and fluidity in the plane of the membrane. Yeah? So this is a dynamic process. Okay, so if you look here, okay, this shows the components, yeah, the organization or the structure of the plasma membrane. Okay, you can see here the phospholipid, where the yellow color here, these are the main component of the plasma membrane. Then you have the cholesterol here. Yeah, so the cholesterol, it lies between the hydrophobic tails, yeah, of the membrane phospholipid, and it also have the integral proteins. Okay, so the integral protein here, if you look this integral protein, it extends through both layers. Okay, there's also another integral. It may have other integral protein that may not extend through both layers, right? Okay, so and then another proteins are the peripheral proteins. So if you look at this, the location of this peripheral proteins, it is on the inner or outer surface. Okay, so if you look here, these are the extracellular side of the membrane, right? And here is the cytoplasmic side of the membrane. So the peripheral protein can be found here, okay, the cytoplasma side of the membrane on the surface of the phospholipid bilayer, okay, or the outer surface, the peripheral proteins. And then you have another components, what we call as carbohydrate. So if the carbohydrate is attached to the proteins here, okay, so it will call as the glycoprotein, right? Whereas if the carbohydrate is attached to the lipids here, right, it will call as glycolipids, right? So, so if you look here, okay, the diagram here, what makes the plasma membrane so special? Yeah? So special. Okay. So what makes it special is its fluidity. Yeah, fluidity. It has make it a perfect balance. Yeah. So the substance can easily across in and out, enter and going out the cell. Okay, but it's not two fluids. Yeah, and they have many components, right? The phospholipid, cholesterol, and proteins to keep all the cells together. Okay, together in plant cells, in the plant, and also in the animals. You know that plant and animals, they have trillions of cells. Yes, so this what keep all the cells together. All right. So now we look at the structure of plasma membrane, the fluidity of membranes. Okay, as I mentioned just now, that the membranes are not static; they are dynamic. Yeah, they move. Yeah, they can move, right? Yeah. All right. So what are the factors that affecting the fluidity? Okay. So it's listed here. There are three factors. Okay. One is the movement of phospholipid molecules. Okay. So these are the phospholipid molecules, right? Then the presence of unsaturated fatty acids, and the presence of cholesterol. I will explain in detail for each of these factors. So the first factor is the movement of phospholipid molecules. Okay. So this movement of phospholipid held together by the hydrophobic interactions. Okay. So in the phospholipid molecules, they have two types of movement. One is the flip-flop movements. Okay? Flip-flop means the phospholipid will move across from one layer to another layer. Whereas there is another movement, what we call as lateral movement. This is within a layer. Okay? So you can see here the arrow here. Yeah, shows by the red arrow, right? So flip-flop is only occur once per month, whereas lateral movement, it can occur 10 to the power of 7 times per second. Yeah, right. Okay, and now we look at the slides, right? The temperature. Yeah, the temperature have the effects on the phospholipid movement and their arrangement. Okay, so when the weather is cold, okay, the phospholipid are found closer together. Okay, whereas in a hot temperature, right? You can see here in the diagram. Okay, the phospholipid arrangement, they are further apart, right? Okay, so this second factor is the presence of unsaturated fatty acids, right? So if you look here, okay, the phospholipid, right, it has a straight tail here. It has two tails. One is the straight tails, okay, And then the other one is the band tail. Okay, you can see here the band tail here. Okay, the saturated fatty acid here can be found in the straight tails. Saturated fatty acid have no double bonds. They are saturated with the hydrogen. Okay, so they are straight. Whereas unsaturated fatty acid, on the other hand, it contains one or more double bond. So it often result a band, or in some book you can see what we call as a kink, KINKS. So the unsaturated hydrocarbon tails, it cannot pack together as closely as when we compare to the saturated hydrocarbon tails. Okay, this is due to the presence of the double bonds, which form the band or the kinks in the tail. Okay, so here the membrane are more fluid. They make the membrane more fluid. Okay, so you can see here, the unsaturated hydrocarbon tails of the phospholipid, they have the kinks. That keeps the molecules from packing together, so they are further apart. So the membrane will be more fluid. So here, saturated hydrocarbon tail, the hydrocarbon, the phospholipid are more what we call is closed. Okay, so more fluid. Example given here, like the oil, they are more fluid. During this very cold temperature, during cold temperature. Okay, now we look at another example. Like this one, the pictures here shows the winter wheat. In this plant, they can tolerate the extreme conditions. The extreme cold, phospholipid will be increased. Why? So that it can keep the membrane from solidifying during winter. So it's important that the membranes keep what we call this fluid, not solid. Okay, this is another example as an adaptation mechanism where the organism are adapting to cold environments by changing the proportion of the unsaturated fatty acid. In response to the low of temperature. Okay, the third factor is the presence of cholesterol. So where can you find the cholesterol? So if you look here, the cholesterol lies between the phospholipid. It's like a sandwich here. So the cholesterol between phospholipid in the plasma membrane of animal cells. So this cholesterol, during warm temperature, for example, 37 degrees Celsius, it will restrain the movement of phospholipid. So the membrane will become less fluid. At cool temperature, it maintains the fluidity by preventing tight packing and slow down the solidification of membrane. So these are the importance of the cholesterol, right? So here it shows the cholesterol within the animal cell membrane. Okay, it reduces the membrane fluidity by moderate temperatures. This by reducing the phospholipid movement, right? And at low temperature, it hinders solidification by disrupting the regular packing of the phospholipid. Okay. Another function of this, what we call the cholesterol, is as a fluidity buffer for the membrane. So here it resists changes in membrane fluidity, where it can be caused by the changes in temperature. Okay? And then to make the membrane work properly, it must be fluid. The membrane must be fluid. Okay? Why the membrane must be fluid? Okay? If the membrane is solidified, then the permeability changes. The permeability changes, then enzymatic proteins in the membrane may become inactive. That's why membrane must be fluid. Must be fluid to make it more work properly, function properly. All right. So I have a summative question here for you to answer later on. Okay, you can refer to my lecture notes, and also you can read the textbook for further informations. Right. So the first question is, list 3 components of plasma membrane. Okay, can anyone remember the 3 main components of the plasma membrane? If you can answer it now, it's good. Any students? What are the 3 components of plasma membrane? Students, can you hear me? No. No answer? Okay, so list 3 components of plasma membrane. And then we look at the second questions here. Name 2 types of phospholipid movement. Okay. So the first question is list 3 components of plasma membrane, which are the phospholipid, and then the proteins, and also the carbohydrates. Right. Okay. And then 2 types of phospholipid movement. One is the flip-flop movement, and then the other one is the lateral movement. Right? Yes, I see, can hear you. Okay, why is the plasma membrane described as a fluid mosaic? Okay, can anyone answer? Fluid mosaic. You remember the fluid? Okay, I show back the membrane here. Okay, the mosaic. Okay, this one. Okay, the fluid mosaic model, right? Okay. So why do you think it's called as the fluid model? Mosaic model. Mosaic. Have you seen mosaic? In the house, there's a mosaic, kan? You can see the mosaic, right? Flooring, right? Then fluid. Okay, so I want you to try to answer the questions. Yeah. And then another question is on the last questions. Yeah. How an unsaturated hydrocarbon chain maintain the membrane fluidity? Remember, where can you find the unsaturated hydrocarbon chain? The straight tail of the phospholipid or the bent tail of the phospholipid?
Bent tail.
Bent.
Yeah, good.
Band.
Yeah, band tails, right? So how they maintain membrane fluidity? The unsaturated hydrocarbon, the proportion, the ratio, right, will be increased, right? Yeah, it changed. Yeah, remember that I showed just now here, and the presence of the double bonds that form the kings here. So this one, the percentage of the unsaturated will be increased. Yeah, unsaturated hydrocarbon tail wicking make it more fluidity, right? The membrane will be fluid. Okay, so I finished the first part of the lectures, right? So for the next session, I'll continue on the sharing session. Yeah, so I will stop sharing now the slides. Just give some information of my research, right? Okay, for your information, I'm a lecturer working in Faculty of Applied Sciences. Okay, just now, Dr. Masita also have introduced my research area, yeah, on the plant biotechnology, phytochemicals, plant physiology, yeah, plant tissue culture. Okay, and I have some experience in the plant development, yeah, cell and tissue culture, and the production of pathogen-free plants and micropropagation, okay, by the technique of plant tissue culture. Okay, and also I have my interest is on enhancing the phytochemicals, yeah, in plant tissue culture, yeah, through the in vitro techniques of plant tissue culture to increase the phytochemicals, such as the triterpenes. Yeah, so there are few studies have been done, yeah, on the production and the enhancement of the phytochemicals through the technique of tissue culture. Okay, so another information that I would like to share is on my program. Okay, I'm in the School of Biology. Okay, under School of Biology, there are 5 programs. AS201. This is Bachelor of Science, Honors Biology. Okay, so this course, yeah, offered in Shah Alam campus, Arau campus, yeah, in Perlis, Jengkel campus in Pahang, and also Kota Kinabalu in Sabah. Okay, this is Bachelor of Science Biology. So I'm under this program. Yeah. So under this program, there are 7 semesters. Yeah, 7 semesters. The program more focus on the biological principle, yeah, by emphasizing the issues and problem that have an impact on day-to-day activities. Yeah. So it provides knowledge in the field of biology, principle, okay, courses related to it also biology principles, but also it offer courses related to business and entrepreneurships. Okay, so under this program we include the element of IR 4.0, yeah, flexible learning, elective courses, and also MOOC, or the massive online course. Yeah, so you can have a look at the website School of Biology. Okay, so this is for the Bachelor of Science Biology, but under this school also we have Biomolecular Science, but only this is for degree program, only offer in Shah Alam. And then we have biodiversity management that is offered in Jengker campus. And another program is sustainable marine technology offered in the Arau campus. And the last one is the applied microbiology. Now it's offered in Shah Alam. It's a new program, Shah Alam campus. So students, after you have finished the degree program, you have the opportunity to further your studies, either during your research by courses or by fully research. So in Shah Alam, we have offered two programs. Uh, a master in applied biology and also master in applied microbiology. And there are many programs in fully research that you have the opportunity to further your study, okay, and your PhD. All right, so I will stop now, Dr. Masita. Okay, thank you very much.
Okay, so student, we're going to have 5-minute break. Okay, if you want to go to toilet for a while. Okay, and then we straight away to continue after this. Okay, 5 minutes only. Okay, hello everyone, and welcome back. So we have already have a break for 5 minutes. Now, I think Dr. Norrizah will continue with the lecture. Okay, please, Dr. Norrizah.
Okay, so we'll continue with the second part of the lecture, right? So I will share the slides now. Okay, can you see the slides? Can you see the slides clearly?
Yes, Dr. Yes.
Yes. Okay, now we go to the new slides. Yeah? This is about the membrane proteins and their functions, right? So just now, mostly the lectures, the information covers about the phospholipids, right? Yeah, and a few information on the proteins. Okay, so here we will focus on more focused on the membrane proteins, yeah, and the functions. Okay, so we look back at the locations of the proteins. Yeah? So you can see here there are integral proteins, and it also has the peripheral proteins. Okay, so the integral it it span across the phospholipid bilayer. Okay, and the peripheral proteins, they are located the surface of the phospholipid bilayer. Yeah, so peripheral it bound to the surface, whereas integral proteins, okay, it penetrates the hydrophobic core. Okay, so if it is span the membrane, we call it as transmembrane protein. Yeah, so if we look at the structure of the transmembrane proteins, okay, they have two parts. One is the hydrophobic part, which is the X helix secondary structure, okay, and they have the hydrophilic part, the non-helical, where it is contact with different aqueous solution at extracellular and cytoplasmic site of the membrane. Okay, now I'll go in detail the functions of the membrane proteins. Here shows it's listed here six major major functions. Yeah, the membrane protein act as a transport. Yeah, and then the second one is it involved in enzymatic activity, and it also involved in signal transduction, and then involved in the cell recognition, and also as a intercellular joining, right? And function as an function where this membrane protein attached to the cytoskeleton and extracellular matrix. Okay, so I'll go in detail of each function. Now we look at the transportations here. Okay, so if we look at the as a transport protein here, okay, the membrane proteins can allow only the hydrophilic molecules to pass through the cell membrane. Yeah? So transport membrane proteins come in many forms. Yeah? So some, they require the ATP. If you look at this on the right side here, it needs the ATP. Whereas here, as a channel, or the it shows the for protein here is provide a hydrophilic channel. Okay, so the particular solute can just across the membrane, Whereas for this this one, it needs the ATP, right? It requires energy to change the shape, yeah, and actively move molecules and other substance across the cell membranes. Yeah? So the substance can be the glucose or the amino acid. Yeah? With so the membrane protein itself, it can release the ATP to use as an energy source, right? Okay, another functions involved in the enzymatic activity, right? So all enzymes, as you know, they are a type of proteins, okay? So as a result, a membrane protein that is embedded into the membrane can sometimes be the enzyme, okay? And they have this active site, okay? So this active site, it facing outside the lipid bilayer, yeah, outside the lipid bilayer. Okay, and this type of enzymatic membrane protein, it can carry out the steps in a particular metabolic pathway. Yeah? For example, like the breakdown of lactose. Yeah? So they can break down the lactose, the disaccharide into glucose and the galactose to the monosaccharides. Yeah? All right, and then another functions of the membrane protein is as involved in the signal transduction process. Okay, so here you can see here, yeah, the membrane protein here, it act as a receptor. Okay, and it also have the binding site, where this binding site have a specific shape that fits the shape of the chemical messenger. So the chemical messenger here, it could be the hormones, yeah, and hormones will connect to the receptor membrane, yeah, that is embedded inside the cell, embedded here in the phospholipid bilayer here, right? And the hormone can change the receptor protein and cause a specific reactions, yeah, depending on the type of hormone or other substance will take place within the cell, right? So you can see here, right, the receptors, okay, the hormones, okay, specialized by type of cell they are attached to. Each type of cell receive different signal from the body or an environment, okay, and then each of these specialized proteins has a special method of transferring a signal into the cell. Okay, some proteins activate other molecules, what we call as second messengers, which carry the message to the nucleus or other molecules. Okay, another function of the membrane proteins is involved in the cell recognitions. Okay, so another important function of membrane protein is in identification and recognition between the cells. Yeah, between the cells. So this is one cell. Okay, this is another cell. Okay, the particular function is useful in the immune system. For example, it helps the body to recognize the foreign cells, yeah, that may be causing infection. Yeah? For example, okay, so if you look here, these are the glycoprotein. Yeah? So glycoprotein, it is the carbohydrate that covalently bonded to the proteins. Yeah? We call it as glycoprotein. These are the type of membrane protein that carry out the cell recognition. Okay, the cell recognition each other by binding to the surface molecule. Often they are the carbohydrate or the proteins on the plasma membrane. Yeah? So the important during the sorting of cells into tissues and organ in an animal embryo, and the basis for the rejection of foreign cells by the immune system in the animals. And then carbohydrate on the external side of the plasma membrane vary among species, individual, and even cell type in an individual. For example, given here, you know that human have different blood types, A, B, AB, and O. Okay, so in human blood types are genetic. Yeah, there are 4 blood groups. Okay, the blood group are based on the red blood cell antigens, antigen present here, which are the protein on the cell surface that are recognized by the person's immune systems. Okay, so for antigen present here, A antigen, so the red blood cell type is group A. Yeah, so these are the red blood type. Antigen B antigen, group B, group AB, it has A and B antigens. And group O, there is no antigens. Okay, and then the 5th functions of the membrane protein act as a intercellular joining molecules. Yeah, so this is the adjacent cells, the neighboring cells. They can connect in a range of different junctions. Okay, they may hook together in various kinds of junctions. So if you look here, there are 2 types of junctions. One is the gap junctions, where here it provides cytoplasmic channels between adjacent cells. So the functions of these functions, actually it helps cells to communicate with each other and transfer materials between one another. Right? And there's tight junction, it preventing the leakage of extracellular fluid. Okay, the last part. The last part is on the attachment to the cytoskeleton and extracellular matrix. Okay, so it acts as an anchorage. Anchorage. So it becomes point of attachment of the cytoskeleton and the extracellular matrix. So you can see here there are many microfilaments or under element of the cytoskeleton. Okay, may be non-covalently bound to the membrane proteins. So what's the important of this network? Okay, they help to maintain the shape. Attaching the filaments or fibers in the cytoplasm help to maintain the cell shape, and it also keeps the location of the membrane protein become stable. Okay, and then attaching membrane protein to the extracellular matrix can help the ECM, extracellular matrix, to mediate changes that occur in extracellular and intracellular environment. Yes, so it stabilizes the location of certain membrane proteins. All right, so this is the some of the summative questions that you can answer, right? So name 2 types of transport protein, students. What are the 2 types of transport proteins? Can you remember? Transport proteins. Okay. And then the second question is briefly describe how membrane protein involved in signal transduction process. Okay. So remember the signal transduction process just now. So the first question is the channel channel protein and then transport carrier protein. Yeah. Then signal transduction process. Yeah, you can see here. You can read more on in the information in the textbook, right? And then we look at the next questions. Name specialized proteins that are embedded, yeah, in phospholipid bilayer, and describe their functions. Remember the name of the specialized protein that are embedded in phospholipid bilayer. Yeah. So try to answer all these questions. Okay. Okay, I will continue with these lectures on the topic of membrane structure results in the selective permeability. Okay, so now firstly we look at the hydrophobic molecules. Okay, so what's the example of these hydrophobic molecules, which are the non-polar? Okay, example are the hydrocarbons, carbon dioxide, and the oxygen. Okay, so this hydrophobic molecule, it can cross, yeah, it can cross the phospholipid bilayers, whereas the hydrophilic molecules such as the small uncharged polar molecule like the water and the ethanol, okay, some ions, chloride, potassium, and the sodium, okay, and also large uncharged polar molecules, example like the glucose and the sucrose. Okay, so this molecule, they cannot across the phospholipid bilayer, right? So how all these ions and small molecule and large molecule can cross the phospholipid bilayer? Okay, so we'll see, yes, the transport where this membrane structure result in selective permeability. We look at the transport proteins here. So here is the transport protein. They only allow hydrophilic substance, yeah, across the channel proteins. Yeah. So the channel protein can be called as its name is aquaporins. Yeah? The word aquaporins, aqua refers to water, porins is the fall. It only facilitates the passage of the water molecules, yeah? The water molecules. And other transport proteins, what we call as the carrier proteins. Yeah? The carrier proteins, where it binds to the molecules and change the shape, and then it will shuttle the substance out across the membrane. So the protein transport is specific for a substance it moves. Yeah. So the protein, the membrane protein, it produces the ATP. Yeah. It releases the ATP as a source energy. And there are two types of transport across the plasma membrane. One is the passive transport, the other one is the active transport. Passive transport, if you see here, is the diffusion, whereas and the facilitated diffusion, whereas the active transport, yeah, it needs the ATP, the presence of the ATP. Okay, the simple diffusion. Okay, the simple diffusion. Okay, so you can see here the simple diffusions, where these molecules diffuse from where it is more concentrated to where it is less concentrated. Okay, high concentration to low concentrations. Okay, so it diffuses down its the concentration gradient. Yeah, high to low. Okay, so if you look here, these are the molecules of dye. The the water. Okay, so it the molecules, it will diffuse. It will diffuse from more concentrated to low concentrated or less concentrated until it reaches to the equilibrium state. So it leads to the dynamic equilibrium. The solid molecule continue to cross the membrane, but at equal rates in both directions. Okay. Now we look at here, the diagram here. Okay. Each type of molecule diffuse. Here can see here, right? Diffuse down is concentration gradient. There will be a net diffusion of the purple molecule towards the left. Toward the left here. Okay, so you can see until it reach the equilibrium state. Okay. So the simple rule of diffusion, okay, diffuse is process. Okay. Then we look at the osmosis process. Okay, this osmosis process is a diffusion of water across a selectively permeable membrane. Okay, so here water will be diffused. Okay, so it will be diffused from a lower solute concentration to a region of higher solute concentration until it reach a equal. Because the solute concentration become equal. Okay, so this is a U-shaped glass tube. And at the center you can see the selectively permeable membrane where it separates the two sugar solutions. Here, low sugar of solute, or the low concentration of the sugar, or here it's higher concentration of the sugar. And here only the water will move through the across. Why? Because the sugar cannot move through this selective permeable membrane because the sugar molecule is bigger. Okay, and then here you can see here more similar concentration of the solutes. So water move from an area of higher to lower free water concentrations. So you can see here the water molecules will cluster, will surround the sugar molecules. Okay, and this side has more solute molecules, fewer free water molecules, right? Whereas this side has fewer solutes, more free water molecules. Okay, now we look at the different type of tonicity. Okay, what is the definition of tonicity? Tonicity is the ability of a solution to cause a cell to gain or lose water. Okay, so there are three types of solution. There are tonic, hypertonic, and hypotonic. So we look at the animal cells. Example, human cells here, the red blood cells. What happens if the red blood cell are placed in the solution that is hypotonic to the cell? Okay, hypotonic, so water will move in, enter the cell, and then the cell will become swell and burst, rise. This is the normal red blood cell, which is in the isotonic environment. Okay, so here water flows in and out at the same rate. Right? Iso means same. So there is no net movement of the water across the plasma membrane. Whereas if we look at this picture, what happened is we place it in the hypertonic solution, and the water will move in, the cell becomes shriveled, and then it can probably die. Okay. The example effect of the increased salinity of a lick to the animals. Okay, whereas now we go to the plant cell, hypotonic solution. What happened? The cell will become turgid, but it will not burst as what is shown in the what we can found in the red blood cell, because the plant cell have the cell wall. Yeah? The cell become turgid. Just be very firm. Okay, so this is important for the herbaceous plant, herbal, right? So I hope that students know what is herbal plants, herbs. Okay, the isotonic solutions, okay, the cell become flaccid, then wilted, no change in the cell's volume, right? And then in the hypotonic solution, what happens? There's a situation here, the phenomenon what we call as plasmolysis, okay, where there is a net outflow of the water by the process of the osmosis. Yes, so the vacuum here become small, right? Yeah. So the plasma membrane pulls away from the cell wall. If you look here, the vacuole is bigger. If you compare to this, the size here, yeah. So this phenomena we call it as plasmolysis. Yeah, plasmolysis. Right? And this causes the plant cell to weak. Okay. Okay, now we look at the these channel proteins. Yeah, the channel proteins, where it allows specific molecule or ions to cross the membranes. Have hydrophilic passageway that allows the water molecule. Example like the aquaporins. Yeah, so these are the water channel proteins. It facilitates the massive amounts of diffusion of water molecule in the plant and also in the animal cells. Okay, so if there is no aquaporins, what happens? The rate of water movement across the phospholipid might be very, very slow. Very, very slow. So must have aquaporins. Yeah. Okay, now we look at these ion channels. Okay, we can categorize the ion channels into two. One is leak channels, where these channels are always open all the time, whereas the another channels, what we call as gated channels, right? It can open or it can close. Okay, so it open or close in the response to a stimulus. Okay, so the stimulus can be chemical ligand or the change in voltage, the electricity or the mechanical. Okay, so now we look at the chemically gated ion channels here. Okay, this is a non-gated or leak channels here, right? Non-gated or leak channels always open and responsible for permeability, specific for one type of ion, although not absolute. Okay, gated ion channels. Now we look at the ligand gated. Yeah, so here you have the plasma membrane. Yeah, the receptor here. Okay, and it can close or open. If it is open, right? It is closed. Both the ions cannot move across, right? The sodium and the potassium ion cannot across. Okay, whereas here, right, there is a receptor here. Okay, and the neurotransmitter chemical, okay, can attach to the receptor. Yeah, so the opening and the closing of the plasma protein here, right, in response to the ligand that binding to a receptor such as the acetylcholine. Yeah, acetylcholine. And then this is a chemically gated ion channel. Whereas the voltage gated ion channel, here it will open or close in response to a small voltage changes. Okay, so you can see here the positive, yeah, the positive here outside, yeah, is make the plasma membrane here close, yeah, right, and potassium, the sodium cannot move in because this is a close, and the voltage gate is closed. The channel is closed, right? Whereas here is open, right? You can see here the charge here, yeah, is inside here. It's now the charge is outside the plasma, outside at this side, yeah, here inside. Yeah. So it open or close in response to the small voltage changes. All right. So now we look at the we still on the channel proteins. Yeah. So this channel protein, yeah. We look at an example of the disease, yeah, which involve the channel proteins. Yeah. So one of the disease is what we call as cystic fibrosis. Yeah. It is a genetic disease characterized by the production of abnormal thick sticky mucus. Yeah. Sticky mucus. So you can see here the mucus. Yeah. It was produced, yeah, at the respiratory tract here, right? Tracheoles, then the trachea, and it can also block this respiratory, blocks the SX, and mucus also blocks the pancreatic ducts. So if you look here, the chloride transport across the membrane are closely linked to the natrium, to the sodium transport. Okay, so the cells lining the respiratory tract cannot absorb respiratory tract, and it cannot absorb the natrium, sodium chloride here. Okay, so the sodium chloride will accumulate. Okay, so it will affect the natural defensive proteins, what we call as defensive, that kill the bacteria in the lungs. Yeah. And bacteria will trigger the airway cell lining of the respiratory tract to produce more mucus. Okay, more mucus, excess abnormal thick sticky mucus, where it will clog the respiratory airways. Yeah? Clog the airways, you make the lung damaged and the respiratory system will fail. Failure of the respiratory system. So you can also read, you can further read on this gene that are involved with this, that involved in this cystic fibrosis. Transduction conductor regulator, or what we call as CFTR gene. This gene associated with the cystic fibrosis, right? So that one is for your further informations. You can read the textbook. And then we look at the carrier proteins here. The carrier proteins, disease caused by these carrier proteins. What we call the disease, it is an inherited disease, cystinuria. Cystinuria. This disease caused by too much of the amino acid cysteine or lysine or the arginine in the urine. So mean that there are too much of the amino acid. Too much of amino acid. And then after entering the kidneys, cysteine dissolves. These are the two kidneys. Cysteine will dissolve and then it will goes back to the bloodstream. This is during the normal conditions. However, people who have this kind of disease, cystinuria, the carrier proteins that transport the cysteine across the membrane of the kidney cells are missing or defective. They are missing, so this will result, the cysteine will accumulate in the urine. So once the cysteine accumulate in the urine, then it will form a crystal, or you can see here, right, near to the these are the kidney, the cross section of the kidney. It will form a crystal or stones, which block the kidneys, ureters, or the bladder. So you can see here, the stones here. It will block the ureter. You can see here, the stones can be found at the lining part of the bladder. Right? Okay, so this is the last slides for the lectures. Right, now I will give you some summative questions that you can answer, right? Okay, summative questions. One is describe simple diffusion. And then what happened to an animal cell when immersed in the following solutions, hypertonic and hypotonic. So you have to read clearly here. This is an animal cell, right? And then name the two types of ion channels, right? So so this is my last slide. Okay, so I hope you will answer the questions and read the books to get more information. Okay, so that's all for today, right? So I will stop sharing the slides. Back to the moderator, Dr. Masita. Thank you very much.
Okay, thank you, Doctor. Okay, now we open for Q&A sessions. Okay, you can directly ask questions. Okay, just turn on the mic And you can also type your question in the chat box. Any questions from the student? Just turn on your mic. You can ask the question regarding the plasma membrane transport. Dekat YouTube dah.
How many students for this semester? All of them are online, right? Is it in the campus
half,
or
online.
at home? Half at home and half in the campus.
Bukan dekat half in the YouTube channel, watching through YouTube channel, and half in this Google Meet.
Okay.
Any question?
Okay, if there is no questions, right? So I would like to thanks to all the students for attending my lectures, right? So if you have any questions, you may ask me through my WhatsApp. My WhatsApp, my number is 013-2626-737, right? And I would like to thank to the moderator also, to Dr. Masita Abu Kasim, and to all the lecturers, Dr. Kaniza, Dr. Masani, and the rest. Okay.
Yeah. Okay, Doctor.
Okay. Yes.
Before we end the sessions, before we say goodbye, do you have any advice for all the students in this Pusat Asasi Dengkil?
All
right.
right.
To give
Okay. I would like.
them.
Okay. Okay. How many of, maybe they can hear my questions now. How many of you would like to become a doctor? Most of them are going to become a doctor, right? Medical doctor, right? Right. So, okay. So for those who are, either you become a medical doctor, okay, or other courses later on, okay, you need to study hard, right? Okay. You need to really focus on your study, okay? Understand, not only memorize the facts, yeah, but also need to understand the process and the mechanisms, yeah. So you know that there are so many process mechanisms in the animal cells, in the animals, in the plants, yeah, and also in the microbes. Okay, so for those who don't want to take the medical courses later on, right? We welcome, yeah, the students who are willing to join Faculty of Applied Science. As I mentioned just now, there are 5 programs that you can choose, select. There are 5 programs offered by the Faculty of Applied Sciences, and we have many campuses throughout Malaysia, right? So you can choose the Biology program, Biomolecular Sciences, Biodiversity Management, Sustainable Marine Technology, Applied Microbiology. Okay, so biology is very, very interesting areas. Okay, and later on, you can become not only the researcher, the scientist, okay, the technologies. Also, you can become lecturers as such as Dr. Masitah, your moderator, right? And also can become an environmentalist, right? And then in marine technologies, there are so many prospects, yeah. And also as a microbiologist, okay. So for the time being, I would like to advise all of you to really study hard, concentrate, not concentrate on your study, right? Read more books, get more knowledge, and discuss with your friends. That's all my advice. Thank you very much, Dr. Masita. Right?
Okay. Okay. Thank you for the wisdom words. Yeah. Okay. I'm sure students will be feel more motivated after this. Okay. Thank you for the lecture today, Doctor. Okay. We are Doctor
Welcome.
already clarifications on and sharing about the plasma membrane transport, and also the sharing sessions about the faculty science gunaan. Okay, and then the experts, the expertise from your side. Okay, and to students. Okay, please, you can find attendance links in the chat box, and the QR codes also will be displayed on the screen shortly. Okay, so as a saying goes, to every beginning, to every beginning there is an ending. Okay, now the lecture has come to its end. Once again, thank you everyone for gracing our lecture with your presence, and we are truly appreciated it. Okay, I end my duty as a moderator for today's lectures with wabillahi taufiq wa hidayah. Wassalamualaikum warahmatullahi wabarakatuh, and goodbye.
Okay. Thank you. Thank you very
Thank
much.
you.
Waalaikumsalam.
Doctor.
Thank you, Dr. Masita, Dr. Kaniza, and the rest. Yeah?
Thank you, Professor.
Thank you, Madam.
You're welcome.
Thank you, Madam.
Thank you,
You're
madam.
welcome. You're welcome.
Thank you.
Now I can see your
you.
face.
Thank you,
Now I can
madam.
see your face.
Thank you,
Open your
Professor.
video, please.
Dah habis baru nak buka.
All right.
Bye, madam. Bye, madam.